everybody, welcome back. So today what we're going to do is kind of extend off of the genetics that we learned last time. So if I kind of go back before break, we did some genetics problem solving. So your task was to pick, let me go ahead and actually hop in and show you. Remembering all the way back to what we did last time, this is not the slide that I want, so just hang in there for one moment. You kind of had a choice. So you could have picked the kind of simpler if you wanted some more basic practice, the harder for middle of the road, and then hardest. And so before we left for break, your task was to kind of pick one of these and work through the genetics questions using Punnett squares to help you and submit those to Schoology. So if you haven't done that yet, that's one piece that you should make sure to have complete. Today what we're going to do is kind of branch off that a little bit, and we are going to focus in on blood typing. If you remember, I showed you a video about kind of exceptions to the rule about our basic Punnett squares. Blood typing is one of those exceptions to the rule. So I'm going to go through this presentation now, which is what we would do in class, and then there is a sheet at the end of the presentation for some practice problems, and that's what I'm going to have you guys complete on your 18B page. So let me hop right in here. We are going to learn how blood works. So this is going to be its own separate fresh entry, entry 18. So please take out your notebooks, open up to a fresh page. Our essential question today is, what is blood type and why is it medically important? So we're going to learn about it, we're going to learn some facts about it, and then we're going to do some genetics problems with it. So this is kind of a combo lesson about the awesomeness of blood and also the genetics behind. All right, if you didn't get quite a chance to write that down, go ahead and pause and get the essential question down before we go on to the next slide, and then push play. Okay, so the idea here, and underneath your essential question, I think it would be good to just kind of write this down as a fact because it pertains to blood groups specifically. Some genes, and remember, a gene is a piece of DNA that codes for a protein, are controlled by just more than 2 alleles. So we get one allele from mom and one allele from dad, and we've kind of learned up to this point that they can be dominant or recessive. And the dominant one masks the recessive one, and there's kind of only 2 options. And your combo of those 2 things determines your genetics. But some genes are controlled by more than just 2 choices. For example, blood types are actually controlled by 3 different alleles. And so those 3 different alleles, 2 of the 3 are co-dominant. And I will explain what that means in a little bit. But those 3 alleles, we'll call them A and B and O, then convey a total of 4 different blood types. So as a blood type, you could have A, you could have B, you could have O, or what we will then learn also is that you could have AB. So there's kind of 4 different major blood types that a person can have, and it's a result of having 3 different allele options. Now in here I said that 2 are co-dominant. So I want to take a quick side note and have you guys remember what co-dominant is. It was in the video that we had watched before break, but we haven't kind of taught into it. And you don't necessarily have to write this down other than to just refresh your brain. Co-dominance, co means together. So that means 2 alleles are equally dominant. So one is not able to mask the other. When you put the 2 together, they essentially express together. So for example, if you have a red cow and a white cow, and the red and white alleles are both co-dominant, you end up with what's called a roan cow, where it shows up with both colors. In flowers, if you have a red and a white flower, co-dominance would then have, both of the two alleles expressed in the offspring. So that's when we end up with these multicolored flowers that are both red and white. So that's what codominance is. And when we apply it to blood groups, we will see that A and B are codominant with each other. But we'll take some notes on that in a minute, so don't worry about that sinking in just yet. When we are in class, I am going to show this quick little video clip about the amoeba sisters. So it talks about multiple alleles, and it specifically uses blood groups as an awesome example. So if you are not in class and watching this, make sure you go to the PowerPoint and just give it a view, or search it up on YouTube. It's a nice refresher that will help drive home some of the points about blood type that we're going to learn in a minute. Okay, so what we're going to do right now is I'm going to give the class in real time a couple of minutes to set up a chart on the 18A page. So we're going to be filling this in as we go through the notes, but it's just kind of easier if we set up the table now and then just kind of have to fill it in. So you can see along the top of the table we have type A, B, AB, and O. So those are kind of the four major blood type options. We'll learn about positive and negative too in a little bit. We're going to do a picture of the blood cell. We are going to talk about an antigen, which I will define in a moment, and then the genotype, which is where our genetic combinations come in. So I'm going to ask you to pause here, recreate this table in your notes, and then when you're ready to move forward, go ahead and push play. All right. So when we're talking about blood types, blood types are determined by specific proteins, which are called antigens in this case, that are located on the surface of the red blood cells. For ease, we're going to call red blood cells RBCs. That's kind of the common abbreviation for red blood cells, and it just makes it a little bit easier. But a blood type is kind of determined by which proteins are just kind of hanging out on the surface of your red blood cells. And so they are called antigens. And you can have either A antigens or you could have B antigens. You could even have A and B antigens, like this particular blood cell here. Or you might end up with no antigens. There's kind of four different possibilities. So I'll actually kind of highlight this point. I want you to think of antigens as sprinkles on a donut. You either have A sprinkles, B sprinkles, both, or none. And when you have none, that's where we fit into that type O blood type that we'll learn more about in a minute. But that's what I want you to think of as antigens. They are on the surface proteins that denote which blood type the cell is based on whether or not they're there or not there. So what I'm going to have you do is I want you to pause the video at this point and copy these two things rows into your chart that you just created on your A page. So you can see we have a blood cell, and then on the surface we have these antigens. So for this particular picture, purple circles are the A antigen protein on the outside, and so you can see that here, antigen. We also have the B antigen, which in this case is a green diamond, and you can see that. If you have type AB blood, you actually have both, a mix of the two. And if you have type O blood, then you do not have any antigens. So again, pause the video here and get that down in your notes. And then when you're ready to move on, go ahead and push play. All right, so just a little bit of extra information. If you're curious, if you know your blood type, that's awesome. It's actually good to know your blood type for, medical reasons. If ever there's an accident or something, you can, say what your blood type is, but I thought it was also kind of cool to show you the breakdown. You'll notice in this blood type column that there are people that are positive and negative for the different blood types. And so I did want to highlight the fact that the positive or negative is due to something called Rh. And that's a different antigen that's either on the surface of blood cells or not on the surface of blood cells. So if it's there, you're positive, and if it's not there, you're negative. And so we're not going to dive into that, but that's just where the plus and minus come from. It's a different antigen that likes to hang out on the surface as well. You either have it or you don't. And so you can see the breakdown. The most common blood type that exists is O positive, followed closely by A positive. And then you can see the breakdown of other blood types in terms of percentages. So AB negative is actually the rarest of all of the different blood types. All right. So the key here now is we know a little bit about blood types and we know how to identify one versus the other, but how do they actually end up with those antigens on their surface, right? Do they just show up? No, it is genetic. So underneath the table, this is where we actually tie it back to some of the genetics that we have learned. So underneath the table on your notes page, I want you to make sure that you get these bullet points down. And I alluded to it a little bit earlier, but in this case, we have one gene, which is located on the 9th chromosome, and there are 3 different alleles. There is a type A allele, a type B allele, and a type O allele. Type A and B are co-dominant, which is why you can see in the picture here that they both say dominant. And the third one, this little O, is recessive. So if there is an A or a B, they are going to mask the recessive O, just like our normal dominant and recessive. The only way we would ever see the recessive allele or type O is if both of your inherited alleles or your genotype was OO. You didn't have an A or a B to mask it. So make sure that you have those 2 bullet points down before we continue. Okay, and so again, I want you to remember, you get 1 allele from each parent. So you don't necessarily have to write this down, but I would encourage you to write down the genotypes in your chart, or actually I think I allude to it later, so you don't have to do that right this second. But you can add these to your chart. So for example, if you have AA as your genotype, well then that is very clearly type A blood. If you have A and O, remember O is recessive, so that also yields type A blood. So these 2 genotypes, AA and AO, both have the phenotype of A blood. Same thing with B, you can either be BB or BO. AB has to be AB. You get 1 of each. They are co-dominant, which is why we end up with AB blood. That is like the donut that has both the sprinkles. And then O has to be O. That is the recessive one. So the only way you can have type O blood is if you have 2 recessive alleles. So even though there are 3 choices, remember, each person only gets 2, 1 from each parent. So your genotype is always going to be 2 letters long. It's just, the bag of letters that you're choosing from has 3 instead of 2. So you pick out 2 of them, and that's what you have. And your genotype determines your phenotype. Another important thing I want to highlight before we kind of jump into the genetics of it all is that from a medical perspective, our bodies do not play nice with antigens that are not our own. And this is just part of our normal immune system. We fight foreign invaders, whether it is viruses or bacteria or whatever it is. We kind of have our immune system running around our body, monitoring, saying, oh, these antigens or these markers on the outside are us. They are from our cells. Yay, everybody's happy. And then if they see something that they don't recognize as themselves, then they attack it. And so that's kind of the basis of our immune system. Which is why we can't just give blood from anyone to anyone. We have these things called antibodies. So if you would like to pause and write down the text here on the left, you totally can, kind of underneath where you've been taking notes. But understand that an antibody is basically a protein in our body, part of the immune system, to help protect from disease, but they're running around monitoring. So if you have type A blood, they are used to recognizing those A antigens. They are not used to recognizing B antigens, and vice versa. If you have type B blood, they're running around recognizing type B as, hey, that's us. And anything that's not that, for example, type A, they would attack. If you have type A and B blood, well, you're used to seeing both, so it doesn't matter if you get A or B, your body is recognizing both as self. But then in the final one, type O, your body is used to not recognizing any antigens. So if some show up, your body's like, whoa, 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 wait a minute, that's not us, and freaks out, which is where these antibodies come in. These are made to fight. So if you have type A blood, you would fight against what you don't have, B. You're anti-B, because B would mean something that's not you. On the flip side, if you have type B blood, you would be anti-A. If you have AB, well, you're good with all, so then you don't have any antibodies. If you have type O, you have both. You don't need to know this. I just wanted you to realize that our blood makes antibodies against the things that we don't have. So we can't just give blood from anyone to anyone, because there is the possibility that some of that donated blood is, has A antigens, and you're giving it to a person that doesn't have any A antigens normally. And so that person's body would then attack the blood that you're giving. So a person can only receive blood that has the same antigens or no antigens, which is why here you can see from the recipient, which means getting, type O blood can only get type O blood. Because there are no antigens, you cannot give them any of these other blood types that do have antigens. So O is kind of the hardest one to get blood for. However, because it doesn't have any antigens on the surface, any of the blood types can take it, because there's nothing to recognize in foreign. So O is kind of known as the universal donor, because any of the blood types can have and receive type O blood. A naturally can go to type A, but it also can go to AB. You'll see that all of the arrows here are pointing to AB, meaning that AB is the universal recipient. Because it already has both antigens, normally the body isn't going to fight any of them. So it can get blood from O, A, B, or AB. And so I copied this chart into my notes because I thought it was a helpful graphic to have. So if you want to pause and get that into your notes, or take a peek at the notes that I've made as a guide, you can do that too. But the moral here is that if you receive blood that has foreign antigens, then your body will try to attack it. All right. So I know that we kind of digressed away from just genetics problems, and here is a picture of my notes if you want to take a peek or zoom in or try to recreate, kind of your choice here. But I do want to make sure to include some examples of Punnett squares before I give you some practice problems. I thought it was important to learn about blood type, and now we're going to hit a couple of these examples. Okay, So what I want you to do is at the top of your B page. So at the very top of your B page, I'm going to say, let's see, 18B. I want you to write down this example, and then we're going to work it out. So go ahead and pause and write down the example, and then I'll work through it. Okay. So hopefully you have it jotted down. I'm going to kind of work through it now. A husband has type B blood, and a wife has type A blood. Both of them are heterozygous. So that is a key word right there, heterozygous. If you remember, heterozygous means that they have one of each. So dad has type B blood, and options for type B blood are either BB or BO. This would be homozygous. Big B is homozygous. So heterozygous would mean 1B, in this case 1O. O is the recessive. So we know dad is BO. We know mom is also heterozygous. So I know mom is AO. She could not be AA. AA would be homozygous. So if I create a Punnett square, and I have this on a color-coded slide next too. But just kind of working it out, you can see here that we end up with a lot of possibilities. What kinds of phenotypes or blood types are possible among the offspring? We can kind of create a ratio. I have one person that's AB. I have one person that's type O, or on the other corner. I have one person that's type A and one person that's type B. So I kind of have a 1 in 4 chance of getting any one of the 4 blood types when I mix these two people together. So here's kind of what it looks like when you put it all together, and I color-coded it nicely so you can see dad and mom. And I have our phenotype ratio. Actually, this would be really genotype. If I was doing phenotype, let's see, ratio, it would be 1AB, 1A, 1B, you would designate the type instead of the 2 letters. 1 type B, 1 type O. There we go. That looks a little better. Excuse me. And so basically then you would have a 25% chance of having each one of the different blood types. So make sure that you have a chance to get this down because you will be doing some practice problems that have this. All right, let's try another. So here's example problem 2. Again, we're doing this on our 18B page. I want you to write this down and then I'll work it out. So here, hopefully you have it down, and if not, pause to make sure you do. But we have a baby, and I know that the baby has type AB blood. Dad, if we are thinking it's dad, has type O blood. So if they have type O blood, we know that this is recessive. So the only way that a person can have type O blood is if they have 2 O alleles. Can the man be the father of the baby? So we don't actually know what mom is. No idea. But if I were to set up a Punnett square, I don't know what mom is. If dad is hypothetically type O, that would mean that dad would be giving an O to each one of the offspring. Which begs the question, if the baby is type A, B, there is no way that the dad could be type O, because the baby with a type AB blood would need to have gotten an A allele from one parent and a B allele from the other parent. And if a father is type O, they don't have either one of those to give. So the answer to this question is that no, the man cannot be the father of the baby. There is no way a person with type O blood could have a child with type AB. So however you want to write it, if it helps to kind of pause this and get the box in yellow down, you can, but you don't necessarily have to. In this problem, we were still even able to solve without actually knowing anything about the mother. We just know that it's not possible for a dad with type O to have a kid with AB. Okay, so hopefully those two practice problems will help. I'm going to have you guys work on some more practice problems, kind of continuing down your B page. So I do have a worksheet linked out here. You must work through problems 1 through 5. If you want more of a challenge, you can continue on. There's 10 total problems. If I click on it and open it up here. If you have access to a printer, you're more than welcome to print it out. Otherwise, you can just excuse me, copy these problems into your notebook, and that's totally fine too. So I have 5 problems here, and it's basically just asking you various questions, like if a woman is homozygous for type A, again, pay attention to that homozygous versus heterozygous, and her husband is type O, and so on and so forth. So everybody must try the first 5, and then the other ones are optional. I'll put that here too. I will also make it a little bit easier while we're doing this, in the middle of the video, of course, to give a little more space and room. That did not go well. What happened? I don't know. So if you, I have an answer key posted too, so that you, there we go, so that you can see it if you need. That way it's a little bit more clear. And that's what we're going to be spending the rest of the class period on, just giving some time to kind of work through either old genetics problems or these blood typing genetics problems, whatever you need. I have some additional videos and reading if you want to learn more about non-Mendelian or exceptions to the rule of the basic Punnett squares, and then some videos there on blood typing. And to recap what we've done, we've learned a little bit about how multiple alleles work. We learned specifically about blood typing and kind of how blood works. And then we worked through a couple of sample problems. So your job is to just finish up the problems on your 18B page. And I will probably have you upload a picture to Schoology. All right, that's it. Thank you.